Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. We invite you into the stories, struggles, and breakthroughs shaping the future of human health. From cutting-edge brain science to discoveries transforming how we heal, adapt, and thrive, we explore the ideas that matter and the people behind them. Together, we look across diseases, technology, and research fields to understand not just what is changing, but why it matters for patients, families, and the future we all share. Join us as we uncover new insights, spark collaboration, and illuminate the science that can improve our lives. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the future of health science. Joining me today is Dr. Jessica Rexach. She's a clinician and a scientist at UCLA, and she's also a 2024 Toffler Scholar. So welcome, Jessica. So glad you're here.
Thanks so much, Deb, for having me. This is my pleasure.
I'm looking forward to this conversation. I would love to just know, let's just start with your journey. How did you get here, and what was it about the dementias that you really kind of wanted to focus on?
So I suppose you could say it's been the journey of a lifetime almost, because once I got inspired in this space, I just couldn't get it out of my head. So I've been mobilizing towards trying to do something about this problem since I was maybe about 13, 14 years old. So without giving too much away, it's been a long, long road. And around that time, I'm a very thoughtful person, many of us are, and I was just contemplating, what it means to be alive and what it means to be a human being, and had all of these very romantic notions about it. And here came my encounter with Alzheimer's disease in my grandfather, who I was not very emotionally close with beyond him being a grandfather. Because he was always this very career-oriented, not much of a family guy. His identity completely defined by his professional life and his successes. But you could imagine a real sense of identity that he had, and probably because of his dementia, he showed up in our home, and so. Here I was seeing somebody who my whole life was celebrated as a World War II hero. He set up the first Army tent hospital in the history of U.S. military as the commanding officer. So the tents that you see in MASH, they came out of World War II, and that was my grandfather's work. And then beyond that, trained with the Mayo brothers, was the first surgeon of Henry Mayo's son. So, from the medical, from his very storied and successful career, French Legion of Honor, routed food to Paris during the war and saved starving people. I mean, and I see this man wandering through my house. So juxtapose the experience of this robbed identity from a man who no longer even knew where he was and sat down to watch an SNL skit with all seriousness. The Weekend Update was on, and he was so concerned about what was happening in the world, but it was clearly a farce. So that really threw me. And I thought, what a unfair experience for someone to build a life of stories and lose them, to build the life of an identity, the thing that you kind of envision you can hold on to, your relationships with people, and then have to walk through this world without them. So I wanted to do something about that. It really bothered me. And I also was bothered by the need to kind of dignify that experience somehow, So anyway, I didn't know what I was going to do with my life. Again, I was a bit of a romantic. I went to college. I was very scholarly. I just wanted to learn everything. I, sucked in physics. I sucked in advanced math. I was always in the library. I went to Cornell, had a wonderful time, but all I wanted to do was learn. And then I had to apply, I had to have a career out of this, and I didn't have much of a compass. Thought about medicine, but it felt too commercial, for lack of a better term, and I could see the direction of that becoming increasingly challenging to be more of a, an academic, which I ultimately am. And then I experienced scientific research, but I needed it to be anchored somehow. So I learned about physician scientists and started walking that road, and really wanted to learn how to become a professional problem solver. So came to UCLA Caltech physician scientist program, with a lot of passion and romanticism about the experience of learning how to make new knowledge, and had opportunities to train with exemplary scientists at Caltech and UCLA. We did work in chemical biology. We identified a way of visualizing a modification of signaling in neurons that nobody else could see before. And I got to do the first study of how that affects the human brain and affects learning and memory. And it actually is the target now of a drug in clinical trials for dementia. So it was exciting to be part of that. We actually made an animal model to demonstrate that this molecule protects neurons from dementia type processes. So it was exciting to be part of that. And the toolbox already at that time was so enormous for being able to problem solve. And the scientific, go ahead.
Can I ask what kind of what time frame is this for?
I was in my 20s.
You were in your 20s, yeah, because it's amazing. Like, you're 13 and you're driven, and now you're in your 20s, and this is several years ago, I would imagine.
It's almost 20 years ago.
Yeah. And you're in your 20s, and you've already you're already in the drug discovery, and so okay, so that's good. It gives us kind of an idea of the time frame here too.
Yeah, and so because this is not something you just decide to do overnight. The techniques and the training that it takes to get where I am now is, like I said, it's like a lifetime, and you just keep learning and learning more. It's not a switch that turns on and turns off. So at that time, it was just incredible to me, the ability to engineer, with chemistry, to explore phenomenon of the brain. And I was challenged to try to figure out how to sync this with my experience as a clinician, because I'm making all of these observations from patients and have the capacity to do that. But the experimental neuroscience world at that point was somewhat removed from human data, human experience. And so that's where the journey of the last more of decade has been in taking advantage of huge progress in science to be able to allow us to make incredibly precise, complex molecular observations directly out of human, and then model them and integrate them with the experimental systems to be able to use human data to inform cutting, the most complex experimental science that we can do. So that's what we do now. And the journey of where the Alzheimer's disease field has tracked along that career that I've had already has just been absolutely enormous and incredible to participate in. Because in the beginning, when I was at Caltech, we had a couple of proteins and a few genes, and that were very rare. And then, when I came back to finish my medical training, that gene space, the genetic space, just expanded enormously because of efforts to understand the common genetic modifiers that influence this disease. And then now where we sit, I fundamentally say this, and I don't say this with any kind of, I say it with awe. Everything that we need, we have to solve the problem in terms of the science and the capacity to solve the problem, and the only thing that we need that's not guaranteed is the commitment and the doing of it. But it's been incredible to get here as a field where I just saw one challenge after another challenge getting knocked down, as the career progresses, and now we're in this very incredible moment.
Yeah, that's exciting. So what were those challenges? And I'm really kind of fascinated with the comment that we have everything that we need to solve this problem. That, I'm assuming, that's understanding, that's data, that's policy, it's, it's regulatory, it's all those things that have had to get to this moment. Can you kind of, give us a little bit more of your thoughts about what those challenges were and where we're at?
Yeah, so I think we can take it on layers like you just described. From the very focused and scientific layer, one could make a laundry list, but just some examples. When I came to start my postdoctoral fellowship, there was no cellular model of neurodegeneration. So imagine cancer, where we can have an idea about how something works. That's not how science goes. You don't just have an idea and you solve the problem. You actually have to go and prove that that idea is right. You have to construct the thing that you think is causing the problem and see the problem arise from it. You have to prove it. And so you need some way of proving that. So in cancer, we have these cells that just grow like cancer. They are cancer cells. They grow like crazy. And you can go, oh, I think cancer grows because it likes to bite, this special kind of sugars are important. Then you get rid of those sugars. Oh, they stop growing. You've proved your idea. You know what I mean? We didn't have that for degenerative diseases, period, 15 years ago. We have the capacity now to take human cells and build cellular models of the memory part of the brain with circuits that oscillate like the human brain circuits. I mean, problem has made incredible progress, okay? When you think about the human brain and its complexity, one of the challenges is that there's a lot of specialized cells, and we were trying to measure them in aggregate and losing the signal. Well, we can literally measure one cell at a time, one molecule at a time. Now we can understand how these cells are talking to each other, how they specialize in different parts of the brain, how different diseases change them. At a level, I would never have even, if I had claimed that we could do that 20 years ago, people would have thought I was crazy. And here we are with that toolbox. We can speak to each other across the world with our data and see, oh yeah, you have that. Well, let's see if my data shows it. Not just ideas, but actually, oh yeah, there it is. Aligning our work and being able to prove things beyond one group, one observation. Which is what you need to really be rigorous. So just the tools that have emerged and their ability to push the science forward are phenomenal. But that's met with, we had a lag in observational data that we're actually beginning now to crest, although there's still work to be done there. We've gotten really good at it, and there's some key interesting things that need to be profiled, but we had this enormous lag. And now we have to push that.
Can you explain what observational data is for the person that doesn't know what that is?
Just observations, what different types of trajectories are there to getting this disease? How many types, what kinds of brain cells change? How do they change? Where does disease actually manifesting? A whole person's brain doesn't get Alzheimer's disease. Certain pockets, and not the whole thing, certain parts within that. We've now described that at molecular and cellular resolution. This is phenomenal, but we don't understand it all. We have ideas. We have to go do the function and mechanism. So we have now all this observation, and we need to push that forward into that proof of how things work and an understanding of how things work. So that the work has not been achieved, but the tools are there and the pieces are there to now achieve that work. At the human side, we were doing clinical trials that took 5 years, $10 million, and you still couldn't figure out if the drug worked. Because we were relying on cognitive changes that, there's a learning curve when people take these tests that makes it difficult to use them. And people progress very different rates, and we didn't have a good way of measuring. The entire science of clinical trials for Alzheimer's disease has changed. And what's been an incredible COVID era, game changer is the biomarkers, the ability to actually measure in blood a quantitative, which means the level tells you means something. Is it a little bit? Is it a lot? Well, if it's a little bit, there's a little bit of disease. If there's a lot of disease. We could quantitatively measure what's going on in someone's brain with respect to Alzheimer's disease. We have a couple key biomarkers. More biomarkers are coming. And so we don't have to wait 5 years and, $10 million. We can start to see if things are moving the biomarkers in the right direction and make quick, reliable calls about if a drug is working or not in a way that we can actually pay for some of these things to get done. And we can start doing these kinds of trials in completely new ways. We could take people's medical records and start to, flesh out ideas about what drugs they may already be on that might be changing these things by like integrating this type of stuff. But it hasn't been done at the scale that it needs to be done, but it's all doable. So at the level of care, there's been some great important work on models of care, of understanding that Alzheimer's disease and related dementias are not cancer, because when somebody has them, their ability to navigate the system is fundamentally impaired. So there needs to be like care navigators. We still exist in a society where I am very concerned about anybody who doesn't have a friend or a family member to bring them to the doctor when they're starting to have cognitive impairment, because I can tell you the people I see, someone drove them there.
Yeah.
And and so now we have these disease modifying drugs with the amyloid drugs so we still have a lot more work to do, but there are wonderful social scientists and people thinking about these problems, and at the very least there's been a great progress with these care networks that are now been demonstrated to reduce costs and have opportunities to get reimbursed. And we need to scale this up, but the concept is there, the methods are there, the science is there. But the society needs to buy in and needs to disseminate these things, because we're looking at a precipice of people not being able to go to work because their parents are sick, and that disseminating beyond the level of capacity of our society to keep doing things the way that we have been doing. But there are the toolbox is there, and the individuals, with the right outreach, infrastructure to start to address these problems is there, but the decision hasn't been made to engage in them in the way that we need to.
I want to go back to something interesting, because I've never really heard anybody say that. You said, because of the COVID era, what was it about that time frame? Because you led me to believe that because we were looking for COVID and all of that the unintended consequences was just a maturity of markers, blood-based, test that kind of accelerated for other aspects like dementias. Did I understand that correctly?
Some of it was coincident. Some of it was already in the works, but the kind of work that was funded during the COVID pandemic supported the technology and infrastructure to push that work forward even more. But a very salient example of something that really did get, again, there was a kernel of it. Things don't arise from nothing. I described the journey of 20 years, 30 years it takes to be even ready to have something ready to go. But when that opportunity strikes, things can then really take off, right? And the opportunity is really, money, fundamentally, and opportunity around that. So, and need, a prioritization around need. So when patients couldn't come in anymore to get scans and whatnot in clinical trials, like the need to prioritize, biomarkers shifted a little bit, but there was a bit of a coincidence around that. But a lot of things came out of COVID that I think are going to be really important for pushing those biomarkers into the next stage, for technologies for like, at-home test, right? Can you imagine taking Alzheimer's disease biomarkers into an at-home test? Well, we developed so much technology around that. This is how science works. Technology doesn't stay in its lane. You build that out for X, Y, and Z, and then you find an application for it in a completely different disease. It's part of the reason that the investment is so unique, is that, maybe you guessed wrong here, but there's a home for it in a breakthrough in somewhere else, and then vice versa. Yeah. So those technologies that came around home testing and rapid testing are going to facilitate moving those biomarkers forward even more. And not just the technologies, but the investment in groups who have the capacity to do that, so that it can be big enough and agile enough to take on these important problems. The other component is non-invasive monitoring of people's cognition. And that really got a major boost because we couldn't get the patients in to see psychologists to monitor their drug response. And so digital apps for first clinical trial monitoring really got an infusion of energy and resources. And there were some really wonderful people involved in that work that it really took off really beautifully um, uh, during those pandemic years. And now it's going to become a cornerstone of how we do everything. And all of these things reduce cost. We want to get to the place where we can rigorously and with data consider an idea without it needing to be able to make a drug company so much money that they can invest $25 million into even just beginning to think about it. Right? This is a problem. And when you're thinking about modifiers of aging and healthy lifestyles and maybe supplements, maybe there's a vitamin that's helpful. Well, we can't even consider it if the cost of even getting rigorous data is that high. But if we can use biomarkers and real world data and apps, the costs start to come down. And that allows us to be able to offer people, reliable interventions that they can take to have kind of a meaningful path forward instead of always having to fly blind, which is how most people these days who are considering what supplements or interventions or how, diet, I still feel people are mostly flying blind right now. And I think that's a problem that could be very easily solved.
Yeah, I was curious about that because you see patients and then you go into your lab, and I want to talk about your lab, but on the patient side, patients are coming in or family members are coming in and they're educating themselves, they hear things, I would imagine that's kind of hard to navigate, as a clinician, one, to give them realistic expectations of where we're at. I get it all the time. Oh, I saw something on the news, or I read this paper, we've cured Alzheimer's, and or why haven't we cured Alzheimer's yet? Because it's just, I would imagine that expectation, but then also giving them some kind of practical what you can do now, um, again, to the expectation of what it could be for the future. Can you just kind of expand that challenge for you?
Absolutely. So I try to I try to offer my patients a sense of respect by being very clear about why I can say what I can say and why I can't say what I can't say. So if there isn't sufficient data, I can't give a reliable, meaningful recommendation. And this concept of like understanding how expensive it is to run a clinical trial. What are the requirements? What are the limitations to being able to even think about doing that? I think it's important to recognize that. But if we don't have that level of data, we don't have that level of data. And I've seen and participated in trials that sound like an awfully good idea. And everything looks so right. And then nothing worked. So I fundamentally do not believe I can predict whether or not a clinical trial is going to work. That would be a lovely thing to be able to do more effectively. But I have a sense of humility about it. So if I can't predict that a drug that took I don't know $50 million of preclinical work to develop by the best drug development teams in the world or in the country, with the right patients and et cetera. If I can't predict if that is going to work, how am I going to tell you if a supplement from Vons is going to help? But I do think that with the accessibility of biomarkers, which is not fully realized, but getting very close and can already begin to get applied in research settings, I think we should be able to start to envision moving through some of these ideas more swiftly. It's still not cheap, but I think we can start to think about addressing the more holistic aspects of care. And so many interesting observations. How much are we being impacted by pollution? How much are we being impacted by isolation and loneliness? I mean, it can go beyond taking a supplement, and we are in a position, and people are starting to answer those questions. by applying a large cohort, which means many people, precise observations that take into account things like their genetics and where they live and their other diseases, and can begin to try to model how these modifiers might actually play out in a way that we could think about then offering a reliable recommendation and a way of maybe monitoring whether that thing is actually working in people. So this is not science fiction. This is all achievable, but we'll have to see how the resources and the government, everything, are going to start to facilitate these opportunities. These are not easy 6-month ideas, but these are all things that could ultimately be done. And in the meantime, let's find something better than a supplement. Let's get a drug that actually can make the brain resilient to Alzheimer's pathology, or heal it after we start to remove some of that pathology, make the brain younger. We know there are people walking around with Alzheimer's pathology in their brain, and they are probably sharper than I am in their 90s. There is natural variation that tells us that the brain can do an awful lot in certain lucky people to combat this disease. Can we put that in a bottle? So, there's a lot more that we need to do to discover that, really, really, really powerful, effective drug or combination of drugs. But there's also a lot more that we can do to be more accurate and precise about measures, measures and interventions that are already out there and being explored by people, but currently we just don't have the scientific rigor to be able to offer much in the way of recommendations.
Yeah, I love that, because while you were talking, I was thinking, it was a couple decades ago, it was just it's just a disease of the aged. Yep. It's going to happen, and not much we can do about it. And what you're describing is, it may not be about being aged. It may be the process of aging, and depending on what an individual's journey to getting that age, that may have affected them, or that may have created the conditions. And so if we knew that, maybe that's the behavior or the drug or the treatment that needs to be put in place, maybe even earlier, before the disease even happens.
Yeah. I mean, when you think, this is not something we're born with done. This is something that we're doing just fine. And then the stopgap, is released, and the disease process starts. So whatever was lost, could it be regained? Could we push it back another 30 years so that it doesn't even become a problem in our lifetimes? It's I used to think about the vulnerabilities that were influencing disease in some people and not other people. And it's a very reasonable way of framing things, but I've been increasingly thinking about we have a natural capacity in the human body to heal and restore, and it fails in certain people. And that's why they get disease. I've been kind of thinking more from that perspective, because, even in the strongest mutation carriers, they have 40 or 50 years of doing just fine. And then they can't do that anymore, whatever that is. So, so it's not about an inevitability. It's about trying to harness what we've been doing already, but maybe is starting to slow down, or develop some damage, or needs to be restored, needs to be healed, needs to be facilitated. And as long as we're alive, the body is always trying to heal and restore. But of course, as we age, our knees aren't so good. Our skin starts to not be what it used to be. I mean, we can see aging on our entire system. And of course, that's the most important modifier of degenerative disease. So, thinking about that framework and thinking about how we can start to improve our body's capacity to heal and restore and regenerate, might be a helpful way of going about this. And at least is how we're thinking about it. And I think, many other people are thinking about it too.
Yeah. So let's talk about your lab, because you mentioned, you're starting to kind of really think about that resilience, or those questions around resilience. Describe your lab. What are the big hypotheses that you are really focused on trying to, address, prove, disprove? Give us kind of a day in your lab.
Yeah. So we do two things. We have certain frameworks and hypotheses that we're pursuing, but we also try to create resources that we think are valuable for the field to address gaps. So, because the data science part of our lab, we feel we're very good at profiling and describing and curating human brain data from different diseases in ways that are valuable. So we recently did this with tauopathies in a huge collaborative project. Tauopathy is one of the proteins in Alzheimer's disease, but it's also in other diseases. And so we did a comparison of all of them and understanding how the brain changes in those diseases and how it relates to the difference in like the genetic architecture that drives them. So we're very good at that. So we're applying those tools to understanding co-pathologies with vascular dementia and Alzheimer's disease, which is one of them. It's the most common combo in later aged people. We're doing an analysis of young onset Alzheimer's disease, including strange presentations that start in unusual brain regions to see, hey, are our ideas about how this is happening going to hold up when we look at cases that are a little bit of the exception? And then we're looking at resilient brains, brains of individuals that have accumulated disease pathology, but the brain's looking great. Their cells are there, the synapses are there, and the patients were, sharp as a tack when they passed away, even in their 80s and 90s. So we're trying to profile and catalog those cells because we think that fundamentally all science begins with observation. And we want to capitalize on these techniques in cellular molecular profiling that have been recently discovered and continue to get better and better to fill those critical gaps that have always been curiosities of mine in the clinic as well. When I see these patients, why does this person have such an unusual presentation of their Alzheimer's disease? Starting in a totally different part of their brain. Well, we can begin to at least make those observations. But similarly, we've identified what we think are a couple of themes around resilience, around the genetics of different diseases organizing around two phenomena: the most vulnerable cells in the brain and aspects of their vulnerability in response to stress, but also cells that have the capacity to fight disease and in response to stress, and the genetics influences how well they can fight. So there's two different, it sounds similar, but it's actually two different things. One of them is like there are cells that are already kind of predisposed to maybe not reacting very, very effectively because their fundamental molecular pattern relates to vulnerabilities from the genetics of that person. And then there's a second one, which is these reactive cells that are supposed to go clean up damage and fix damage. This is the immune side of our work, that we think that there's actually very specific ways that they're very specific stresses that they're reacting to modify the outcome. And we want to now, test that and then use those systems that we're building to identify new ways that follow human genetic variation to be able to give cells more resilience to disease. So we're really everything, like I said, my framework is resilience, but we have identified contexts that we think that resilience is most important in, that we're trying to model and validate, and molecules that are actually making the difference in those contexts. So
can you clarify the immune system part? Because, people hear about the immune system, like chicken soup and, vitamins and, be immune. But you're talking about a very different immune system, I believe. Is that true or not true?
So our lab is fundamentally, with all the crazy stuff that we do, focused on, have a genetics focus. So we're always trying to understand how human genetic variation is making it so that somebody gets Alzheimer's disease and somebody else gets Parkinson's disease and somebody else does great and doesn't have any of these diseases. And we're trying to understand like how those genetics, what is the actual thing that they are doing in disease in a human that's making that effect, that's taking that effect. Can we then
at the molecular level,
and then can we recreate the beneficial things and get rid of the harmful things. So interestingly, the most variation we have in the human genome is in our immune genes. Not surprisingly, because we have this interesting coevolution with pathogens, where pathogens are evolving very quickly and we're evolving, and like there's the relatedness between the pathogen evolution timescale and the human evolution timescale, like really create these interesting pressures for the diversity of these immune genes. We're actually favored to not all have the same immune profiles in our species, right? So if a virus suddenly finds a weakness, we all get wiped out. So we have a lot of diversity there. And one of the projects that we're just wrapping up is like mapping that diversity relative to every dementia that, and saying, are there specific immune genetic factors that are contributing to why some people may get the disease earlier or faster or just more likely to get it versus others. So we're doing that kind of work, but we're also looking at the brain resident immune cells, which really respond as like a modifier of tissue health. They respond to damage by cleaning it up. They'll signal to the damaged cell to tell it to kind of either reprogram and restore, or maybe quiet down for a little bit. Like they completely orchestrate the way that the tissue damage is addressed. And these cells carry a phenomenal weight of human genetic variation influencing dementia, again because it still relates to the fact that they're already more varied in the genome. So it's kind of the material that our genome has to work with is stuff around immune. But it appears that in Alzheimer's disease, like a lot of what distinguishes the person who gets the disease from the person who doesn't is these type of resident immune cells. They're called microglia, that have to kind of maintain the homeostasis of the tissue as it's injured and restoring. So a lot of our work is on microglia, and trying to capture, improve the ways for us to recreate human microglia, heterogeneity and disease reactivity in systems that then allow us to do drug screens of 320,000 drugs, or genomic screens with the latest and greatest technologies, but specifically organized around those transitions that are key for disease. So we have a lot of things going on that. And then we have more refined work where we fundamentally just understand that there are aspects of how these cells react in tissue that we don't know really necessarily in the big picture. How would they affect cognition? So we've generated a model of the hippocampus, a cellular model, and we're putting in these resident immune cells and seeing how under different states, different stress, different genetic factors, they actually change brain circuits. We think fundamentally anything that we do needs to kind of pass the test of, okay, well, if we make it look better from like the tissue damage perspective, does it start screwing up a brain circuit? So we want to have both of those. So that's where we've been really honing in a lot of resources in the lab, is developing those models so that we can move the observation into something that can be explored in these preclinical models and then developed out into, a drug or an intervention.
Yeah, you said something that kind of triggered a thought about the immune system keeping up with kind of the environment or the things that, we face as humans every day, right? And you brought up plastics, you've brought up stress, loneliness. Is there an acceleration? We all feel like the world is accelerating around us and a lot of changes. Do you think that individuals that are more resilient just have a way to keep up with that change, or then versus people that don't? And I don't know if I'm asking that question correctly, because I'm fascinated with time and change and the pace of change. And it just seems like there's a conflict that for some individuals that just can't biologically keep up with things that are going on versus others that may be kind of fascinating. It's fascinating that their ability is to keep up better with some of the changes that are happening.
I mean, the absolute answer is that the biggest correlates to the resilient tissue, of people who are resilient, it's just that their neurons are intact. And then you don't even see these immune cells cleaning anything up because they're just really quiet. Like the pathology is there at a lower level, but you don't see the reactivity that they have. So, but the problem with like not seeing reactivity is you don't know, is it because they're intrinsically not reacting, or is it because they don't have any damage to react to? And I would favor that it's probably that there's no damage to react to. So, this is where like cross-sectional, when they say like a moment in time of observation is insufficient, and you have to build cellular models to be able to understand the cause and effect over trajectories of time, which we can't do with human, tissue observation. So there's a lot that we don't know about these brain immune cells. They seem to turn over more rapidly than other brain cells, but we don't know what their aging is. Does that? Yeah. So there's a lot of work in measuring the clock of cells, and trying to understand like what's driving the aging. Is it the peripheral immune system, like the stuff in our blood that surveys tissues? There's been a lot of work and interesting work suggesting that maybe just these surveying blood cells are actually driving changes in aging. Or is aging of a brain more about like how well is that neuron doing, and an aging phenomenon, how much virus has gotten in there? I mean, these fundamentally we don't know. But we definitely don't know what aging is of these cells. We don't know, for example, what if you were to take out someone's old resident immune cells in their brain and implant young ones, would it make a difference? Animal study so far suggests it wouldn't, but I'm not sure that that's sufficient. So I think the jury's out about a lot of these questions, and this is what's so exciting. This is an example of what I set up earlier. We make observations, and then we can, not just with like hand waving generalizations, but with some specifics, identify some fundamental neuroscience that we need to address to understand this disease, right? Fundamental neuroscience, and then have an interaction with basic research and translational research in a way that we didn't have before, because we were following the questions from the basic models, but now we can ask these questions from phenomenon of like a resilient brain and say, wait, why is this microglia so quiet, or why are these cells looking bigger? And then try to ask some fundamental things about like just the turnover and aging of glia that we don't know to model it. So this is some of the ways that science is getting really, really exciting.
It's so complex. It's so complex. And for people that didn't grow up around scientists or research, it just seems to be like, wow,
Well, it's like, it's like, I mean, if I had to take my iPhone apart, I would be completely baffled, right?
That's true. That's true.
But this is the phenomenon of specialization as our species like really has gotten quite sophisticated, right? Is that we can't take apart these complicated machines or instruments and be expected, there's 100 things in my office that if I had to take them apart and figure out how to put them back together, I probably wouldn't know how. But when you do it for your lifetime, or that's your profession, like it's just an engineering problem, and so it's hard to convey to others because I describe all of these things that sound so complicated, but it does really feel like we're getting to a point where these things are becoming much more describable.
Yeah.
Where we can point our finger at what we know and what we don't know in concrete, definable, and limited ways, and approach the next stages, always narrowing down the questions rather than only expanding. Of course, you always ask more questions as you learn, but then when you pan out and you say, okay, well, where does that leave us for what I need to get to the next part of my engineering problem, which is to re-engineer human brain resilience or whatever, like. Where is the next piece that we need to specifically get us to that next step? And it does become more precise and less complicated. When I was a graduate student, I thought Alzheimer's disease was a giant black box. Disoriented. We didn't know what was the beginning. We didn't know what was the end. We didn't know what was in it. Now we can describe everything in the box. And we need to understand some, how they relate to one another, where the cause and effect is, and we're making some progress on that, and that's going to start to come together. And of course, that box is the brain. So there's some fundamental things about the brain that as we learn more about brain function, circuit function, memory, which is a wonderful field where we actually have learned. There's like Alcino Silva here did great work on time and how time influences the consolidation of memories, and how, time spans are influenced by brain circuits and phenomenon. So like, the neuroscientists can approach these really incredible questions with sophistication as well. So like, again, you put that all together, and as the neuroscience fields develop, and then the ability for us to describe brain diseases comprehensively is catching up, and then be able to measure them those same phenomena non-invasively in human patients catches up, we're ready to take on these problems like you would an engineer would take them on.
Yeah.
So that's where the potential we've achieved, that we, I think, really need to lock in on, and not lose our handle on, which is what we're at risk of doing if we lose the buy-in for scientific method and progress.
Yeah. Yeah, I think that's really important. And I am encouraged, again, as you were talking, there's, for people that want to know, there's accessibility there's all sorts of graphics out there and 3D models of the brain and the neurons firing, or the cells moving along and connecting and doing their thing. So, I mean, the insight, if people want to get on, TikTok's full of it, or LinkedIn is full of it. You can find that stuff. And I think, and that wasn't there. I wasn't aware of it 10 years ago. In fact, 10 years ago, people in my family, Alzheimer's, dementia, it was all the same, right? It's just, people just, have this condition. So I think, you have described something that's very hopeful, is, where we're at in this moment of time. And I think there's other scientists and researchers that share that. You're still a very young woman, and you're probably looking out 10, 15 years from now. And what do you think, what do you think we'll have achieved? And, where do you think we'll be in that 10 or 15 year mark?
I think that there's places that we can be, and it's a very hard time to speculate on the future. I think I think that a lot of creativity and important and interesting things happen when there's like a state of disruption, and I think that the technology has advanced so much that it's kind of an interesting time to think about having to rebuild some of how we, how we do medicine and how we do research and all of that. And that might benefit from a little bit of a disruption. But I am fundamentally concerned about kind of an over pruning of the momentum in the AD/ADRD research space at this time. And it's just an experience that I fundamentally am in the middle of, because I'm on study section and I'm seeing what I would consider to be very promising grants just not being able to get funded. Not just one, but like many, many, many. And it's, recognizing the investment of time and money that took that investigator to get to that place to ask the next question, and then now we're going to dump them. So I am quite concerned about that, and I think many of us are quite concerned about that.
Do you see the philanthropic environment kind of stepping up to that disruption, or do you? Maybe there's just talk right now, and that might lead to new kind of models for getting things funded. But what are you seeing?
I think that we've had a wonderful relationship with philanthropy, and so I have to acknowledge the people who've donated and contributed, always to science. The scope of what we're losing in terms of federal funding is going to require an enormous recalibration of philanthropic giving to be able to keep up with, and things will need to be prioritized, and we can't make science necessarily cheaper, unfortunately, but we could maybe make it more efficient. But I don't think that that's going to solve the problem. So, so we need to be thinking about, philanthropic giving at scales that would be unique to the moment, and you're seeing pockets of that happening. But I think until it's realized, I just don't know where that's going to be. But at the same time, um, you know, there's there are opportunities to do. So the wonderful thing about philanthropic giving is that it allows for a reorganization of the process. So there's usually less strings attached. When you do something with the government, the grants need to be of a certain style. They need to be very incremental. They require, one group or another group working together. The institutional overhead, all of that is like all kind of baked in. 5-year progress, certain amount of money. But with philanthropy, you can say you could do it however you think it needs to be done. We want to do a moonshot for 10 years, and we want to bring in and fund, the best people around the most important question, and just keep our nose to the grindstone until, or whatever people say, until we get there, and so there's opportunities to do things at different scales, and I'd be very excited to see some of that realized in this moment again, where it's not, if of course I'm looking always for ways to make this work, and this is a life's work, but I see all of my peers and I have a sense of responsibility in the work that I do to make sure we get it done. And so when I see that it can be done by the field, and we may not be able to get it done, it really is very concerning to me. So so I think we need to engage more, and we need to really go for the big goals, and unless there's a concerted big step up, I'm concerned about who's going to be left to do this work in a relatively short period of time.
I was wondering about the brain drain, or the 13-year-old Jessica that's out there, looking at science as a career, or something that she aspired to. You're worried about that too.
Yeah.
Yeah.
100%. And the people who are in it now, it takes so much time to be able to do this work. Again, it's not a switch you turn on and off. And so to imagine building all of that up and then just not being able to execute is a lack of lack of return on a huge investment. And I think there's been good data to suggest that money that goes into science actually is a financial driver, because we can create so many products and so much wealth out of that for the country. But there's skepticism about science. There's I think people don't understand. There have been big reports about science not being done correctly, but all of those things from where I sit are kind of the rare opportunity to think about how can we protect ourselves from that kind of stuff going forward with more transparency, et cetera. So all things, again, problems that we have solutions for now that we didn't have before. I don't see them as a signal that my colleagues or the people that I work with are out there trying to manipulate the world. So all of it is a call always to grow into a stronger, more more effective position. And if you spend the amount of time and energy doing the kind of work that I'm doing, you're not doing it for yourself.
Right.
You're doing it because of a sense of this is something you're put here to do, and you have capacity, you have skills, you were in the right place at the right time, whatever it is, and you just got yourself here, and it's okay, I'm here. How can I serve this problem with all of what I have? So I feel that we have many people in that position, and we need to be very thoughtful about solving the problem we all care about with that opportunity, and not backing down. So
yeah. Yeah, yes, I agree with that. So as we wrap up here, was there a question I should have asked you, or is there something that you want to say that we haven't touched upon? Because, your message of hope and also caution, and also representation of the science community and how it differs maybe from the business community or such, I think is important. But is there anything else that you kind of want to land on here as we wrap up?
Well, we covered a lot, and I, thanking everybody who's listening, because that means they're interested, and maybe trying to find their way of contributing whatever they can to the problem. Maybe it's taking care of somebody, and or just knowing a little bit more, or kind of being part of this energy, which is phenomenal. But I mentioned the word moonshot before. I really do think we should be thinking about this problem now from that perspective, and where failure is not an option. And again, the only reason we will not accomplish this is if we don't put enough into it and we don't do it. And I say that with complete confidence. And I'm not talking about myself. I'm talking about the community people that I stand with. So, so if that can motivate people to try to create the opportunities for that moonshot, so that we can finally go and now systematically, cure these degenerative diseases, so that we don't have the suffering of not knowing your family member, not knowing how to take care of yourself, how to get into your house, affecting huge proportions of our population in a very short order. So if you have the passion or whatever, that just push for that moonshot, because this is what we need to be doing right now.
Yeah, that's great. So, Jessica, how would people get a hold of you if they want to learn more about what you're doing, if they want to, to collaborate with you in a moonshot or fund you? How do people get a hold of you?
Wonderful. So email is great, and we have something of a lab website that's in Renaissance mode. But I can be found at UCLA. But jrexach at mednet.ucla.edu, which maybe we could somehow share, is my email address. And I get a lot of emails, but I do my best. If it's important, make sure to email me again if I don't respond, because sometimes I just can't catch everything in a given day. But absolutely, I want us to be community and engage around these issues. And if there's anything I can do, please reach out. I'll do as much as I can, or try to help direct people to where I think they can get their needs addressed. So happy to do that.
You're wonderful, and I appreciate you and everything that you do, because it's a huge personal sacrifice to throw your life into this, and I really appreciate that.
Thank you. We say as doctors usually how much it's a pleasure to get into the ring with people when they're fighting the fight of their lives. We get a chance to see, I think, some of the most heartbreaking, but also some of the most beautiful aspects of humanity. And that aspect is us coming together around a problem to take care of each other. So it's a pleasure to be able to do that in this way. And, it's all of us doing it together. So thank you for inviting me on, and hopefully this inspires some people to fight the fight with us.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.